Actually, this is our new reality. This could have been a perfect day at the field station, especially with the coffee and the goodies and everything else. But as I mentioned, this is our new reality. So welcome everybody. I would like to introduce Dr. Scott Furlong, Provost, Vice President for Academic Affairs, for some remarks. Scott?
Yep, thanks, Kamal. Satisfy my mute button. I just wanted to say a few things, and I think Kamal's right. I'm missing Rice Creek today. I think it would have been a lovely day to be doing this, although perhaps hard to keep our attention on the great presentations that you're all going to be making. But I want to congratulate you on the research and projects you've been working on. And I know you know this, but the field station is really a unique resource we have here at SUNY Oswego, and I'm sure you've all done much to take advantage of that. And you know, I look forward to this event every fall and to get to see what's going on at the center in the context of research and project-oriented aspects, and seeing what both our faculty and students and staff are working on, and always walk away learning a lot more than when I first walked into the center itself. So I want to again congratulate you. I want to say thank you to both Maria and to Kamal for their work and their support of this event and of all the research going on. And you know, wish you well as you present these projects, and I look forward to hearing about them. Thank you.
Thank you. Our next, Dr. Kristin Croyle, Dean of Liberal Arts and Sciences, her remarks.
Good afternoon. I'm so happy to see all of you here. And to echo Scott's comments, as all of you know, and as all of us know, the Rice Creek Field Station is a real gem for SUNY Oswego. It really is. It's also not a hidden gem. It's one that it's well utilized, that our community and students know is there and come and visit often. It's been kind of sad this year to see people have to be more distant and not be able to come into the building as often. One of the things that makes it such a gem is that it really exemplifies the mission of the university. It encapsulates the things that are best about SUNY Oswego, that we are in this beautiful environment, but it brings together faculty and students and community members to help us better understand the world that we live in and to make it a better place. And the presentations today, I think, are a great example of kind of the diversity of work that happens at the field station and the diversity of the people that it brings together. So again, congratulations to all of our participants on the successful completion of your projects. We're all looking forward to hearing about it today. And yes, thank you. Thank you to Maria and to Kamal and to Kristin and to Wendy for making all of this work together so well. I'm looking forward to hearing from you.
Yeah, actually, thank you to our organizers, Maria, Laurel, and Kristin Haynes. So Maria, it's all yours.
Yeah, so we're gonna do what we came for, is like listening to the amazing presentations. So I wanna just to like tell you a little bit how we're gonna do it. First, I wanna remind everybody to stay mute for the duration of the presentations. And each awardee is going to give a brief introduction of the presentation, and then I'm gonna play the pre-recorded video. And because they are pre-recorded, we're gonna have the Q&A session at the end of all the presentations. So I ask you to, again, please stay muted. And then at the end, you are free to ask all the questions that you want. Now, if you wanna ask the question, we ask you to raise your hand in the Zoom functionality. And whenever it's your chance, we're gonna call you so you can ask your question. If you don't wanna talk, you can use the Q&A functionality that is in the lower bar. So you can ask a question there, and any of the panelists will be able to see your question and will be able to answer the question at the end. Now the panelists are gonna read the question because most of the time you might not be able to see it, and then they're gonna answer. If you are not familiar with Zoom, for those of you that don't know exactly how to use it, but I'm sure most of us know at this point, if your panelist tab or your participants tab is blocking your presentation, you can always like minimize it on the side. So if you see that it's blocking, just click on the small line, gray line there, and that you can minimize it so it doesn't block your presentation. Now with that, I want to first introduce our first speakers that are Cory, Kyle, and Ellen. So they are going to give a brief remark about their presentation, and then I'm going to play their video.
In our presentation, we introduced ourselves, so I'll wait and let that speak for itself. But we just want to thank the Rice Creek Association and Kristin and Wendy, specifically at Rice Creek, for all of the help and the support. And we're really, really excited for this project and future collaborations, really to get students out of their dorm rooms and outside, because being outside feels good.
Hi, we are Cory, Ellen, and Kyle, and we are here to talk about the SUNY Oswego Counseling Ecotherapy Outreach Workshop Series that was held this past semester out at Rice Creek. We hope that you enjoy our presentation.
First, we'd like to introduce ourselves and give you a little bit about our background and our interest in doing this. My name is Cory Brosch. I'm a licensed mental health counselor at the Counseling Services Center. I have a background in ecotherapy, specifically mindfulness-based ecotherapy, as well as trauma work.
Hello, my name is Ellen Gooch, and I'm a licensed clinical social worker. And my background is not only in social work, but I also have a master's degree in therapeutic recreation and ran a lot of wilderness programs out in southern Utah, and just found that there's a huge connection between nature and healing.
And my name is Kyle Dzintars. I'm a licensed mental health counselor in New York State, and I do outreach on campus regarding mental health. And I've always been drawn to expressive arts and connecting to the natural world. So I feel like this was a good fit in some of my interests and what I would like to bring to the campus.
So a little bit about our overview of our project. We wanted to integrate ecotherapy as well as expressive arts techniques into the counseling center's outreach approach. We decided to have a workshop series held at the Rice Creek Field Station. The activities at each workshop will incorporate coping skills as well as foster connection to and appreciation for nature. The project rationale, why we decided to integrate these two things. First, I'll let our outreach coordinator talk about why outreach is important.
Sure. I feel like a big part of why outreach matters on our campus is because I think in terms of counseling services, we want to be as visible as possible to students. And sometimes with stigma and barriers for people entering into counseling services, we look for creative ways to engage with students in therapeutic ways. Rice Creek, I think, offers a natural therapeutic environment. And I feel like in order to help build good connections, help people kind of spread out around campus, see things that they might not encounter on the main campus, I thought it was a nice place to go to get people together and enjoy the natural world. And I mean, maybe experience some kind of calming, grounding experience. So part of the outreach that we were trying and intending to do through this project was to bring people together and experience things outside, especially during this time of COVID.
Definitely. Ecotherapy has been shown to be a multiculturally competent intervention for anxiety and depression. With a diverse population for our students, we really strive for multiculturally competent interventions. Connection to nature is also shown to boost emotional and physical well-being.
So, and connection to campus as to students' functioning and retention. And as Kyle pointed out, especially during COVID, when so many students are feeling isolated, This is an opportunity for students to get out of their resident halls and to come and spend some time out in nature, and to not only feel more connected to the school, but also perhaps a greater connection and grounding with themselves. All right, so the project goals and objectives are, as you can see on the slide, are the following: to improve student connection to other students and the campus, as we've talked about, to provide students with coping skills and strategy to regulate mood, improve students' stress management, improve student connection to nature, improve problem solving and critical thinking skills, and increase student self-awareness and self-reflection. And we hope that through the rest of the PowerPoint, you will see how we have tried to incorporate these goals and objectives even through the COVID pandemic.
So our project plan, the picture's kind of metaphoric. We planted a seedling of this project with great ideas, and we seem to have to plant it in rocks this semester rather than some nice fertile soil. But our starting plan, our little seed for this project, was to have a workshop series for the whole academic year, aiming to have 3 to 4 events each semester. And within each event, they were going to be big events and have 3 or 4 activities guided by the 3 of us, as well as maybe some of our interns or peer educators supporting us. And then to give post-workshop surveys at the end of each semester for the attendees. However, it did not happen that way. COVID-19 hit and we faced some challenges.
Yeah, and so some of the challenges that we did face due to, like what Cory said, COVID-19 hitting, was difficulties getting materials due to shipping and purchasing delays. The Oz on pause due to COVID-19 response, and then the shuttle bus schedule changed.
So we kind of had to be creative, and we still wanted to do this. We found Rice Creek was one of the safest places for us to have students meet. There was so much social distancing, and we wanted students getting outside. We had an opportunity in the beginning of the semester, actually during opening week, where orientation talked to us about offering something at Rice Creek. So opening week for first year and transfer students new to campus, we offered 5 guided walking meditation programs. That went so well, we decided to offer more of that. So we offered 3 mindfulness in nature workshops before the pause hit. So these were guided walking meditations. We had more planned, but with the pause hitting, we were only able to hold 3. We had our big event, our first big workshop that was planned in the project initially was a workshop focused on sounds in nature. We called it Do You Hear That? It had to shift to a virtual workshop involving these sound-based activities. Kyle, do you want to talk more about how we shifted that into a virtual workshop?
Sure. We were trying to find ways to allow students to have a safe experience, because obviously being in person, listening to some of the sounds was our intention. And it was difficult to shift. We had our facilitator, Samantha, come in to do what's called a sound journey, and uses a lot of different instruments and sound making devices to really guide us through this relaxation experience. And it does affect people in different ways. So in order to make that happen, we were able to capture the recording of it. So we do have an audio recording that we've put up on our website that I'll talk more about later. And then we were able to host it on Zoom, which wasn't ideal. And it was a little bit more difficult in terms of quality. But we were able to use even recordings of some of the meditations that we had intended to do in person, and tried to make more of a virtual experience for students to access on their own, which I think opens it up where, I mean, it's something that goes beyond just the set day and time we had intended, and maybe can be useful in different ways for future programming.
And then we also offered a post-election date program. Since we were allowed to have a little bit more in person, we called it Leave It in Nature. That involved self-guided and self-paced activities at Rice Creek for students to come when they were available that afternoon and do things on their own time in their own way, just to help process any stress they were feeling. And then, as Kyle said, we added some guided meditation recordings on our website. We did these on our own that weren't part of the program, where we just went out on our own, recorded some video, some pictures, and then ourselves doing voice recordings of the meditations, compiled them together so they're on demand on the website whenever students need to check in with nature, even if they're stuck in their rooms.
So this is an example of one of the guided meditations, and it's called a nature moment, or NAMO. And it's steps for nourishing NAMO, taken from Nature Creative Field Guide and Journal by the Mindfulness Project. And so if you just sit back, close your eyes if you're comfortable, and let's do a little NAMO moment. So stop, pause, be still, and breathe. Take in the natural world around you using all your senses. Notice how this makes you feel inside. Feel your feet on the ground and sense yourself in this exact moment at this exact place. Take another deep breath. Feel your body centering and carry on. And when you're ready, we'll go to the next slide.
Thank you. Our observations with doing this, first, we were getting students attending these programs that weren't connected to the counseling center through other services. And this doesn't always usually happen for our outreach. So we're really excited about this. In that opening week and then after, we had first year students visiting Rice Creek Field Station, which in the past, I'm not sure some of these first year students with majors that aren't related to sciences, how that they would know Rice Creek is there and get there before this, especially opening week. We also had upperclassmen discovering Rice Creek for the first time. We had students coming to these programs who were like, we never knew this was here. This is wonderful. We had a lot of repeat attendees to programs, so students that might have came during that opening week came back and are looking for it and are following our Instagram, looking for more programs. We had students reporting stress and stress relief and relaxation when they're in the program. And then more of our counselors using Rice Creek as a referral source to go there for students to do that. What's next for us? We don't know. We don't know what next semester is going to look like. We're really, really excited about things happening. We're continuing virtual offerings. That content will be there on our website. We have more planned the rest of the semester, depending how things go. But for next semester, we're really hopeful the stuff that we had planned for this semester we can do. I know I am really excited for some very interactive meditations and walks to get students even more connected to nature on a deeper level. I know Kyle and Ellen are very excited about other things too we're planning.
Yeah, definitely. One thing that I really wanted to make happen this semester that couldn't was using more of the expressive art techniques. And I think watercolors are a really nice therapeutic tool in terms of being creative and having an outlet. And that is something really wonderful to do in nature because you can capture some of the natural world using watercolors. And I hope to be able to find a safe way to do that in coming semesters. And I mean, I look forward to having opportunities to just be, I guess, a little bit more creative with the space because I think people really enjoy being out there. So, yeah.
Yeah, and we're going to just continue to do our NAMO walks. out in the winter, and having people not only appreciate Rice Creek, but also perhaps to appreciate the 4 seasons, especially the winter, and have that just become another experience and a grounding tool for the students that come out and participate with us.
Thank you so much for listening. What questions do you have?
All right, thank you very much for a nice presentation. And our next speaker is Emily.
Hi, I'm Emily. I already introduced myself in my presentation, but I would like to thank Wendy and Kristin for helping me. So yeah. Hello, I'm Emily Cieplik, and I am a zoology senior at SUNY Oswego. And I'm here to present my experiment on color preference of feeders in black-capped chickadees, or Poongodi. Here's a little introduction on chickadees. Also, I'm just going to call them chickadees. Black-capped chickadees is a mouthful, so chickadees from now on. So, chickadees belong to the order Passeriformes and also the family Poongodi. And the order Passeriformes contains over half the amount of birds, half of the species of birds that exist on Earth today. It's like 60%. That's a lot. And the family Poongodi just means they're closely related to other birds like tufted titmice, gray-tits, blue-tits, and there are more birds to the family. It's just those ones are pretty famous. Like we have tufted titmice around here. Chickadees are small and round, and they are constant feeder birds, but you're much more likely to see them at the feeder in winter than in summer, and that's because food is scarce and they're looking for an easy meal. They're omnivorous, which means they'll eat anything from seeds and berries to insects, and in the summer their diet actually consists mostly of insects, but in the winter they have been documented eating off of carcasses, so that's kind of cool. They are non-migratory, which means the chickadees that live in New York, they stay in New York. So here's a little map that shows the range in purple. As you can see, they don't go down south, but they do go up to Canada. They even go all the way up to Alaska, which is kind of cool. And I know I said they weren't migratory, but young chickadees have been recorded traveling longer distances if the population of chickadees is really high. So it's a way to avoid competition. Bird vision. Birds are tetrachromatic. We as humans, we're trichromatic. And all that means is birds have long wave, medium wave, and short wave cones. So they can see the visible spectrum. So this place right here, think you can see my mouse. And they have a fourth cone that allows them to see the UV spectrum. So right here. And the UV spectrum is from 300 to 400 nanometers. And the use of the UV spectrum is actually pretty heavily studied in birds. It's pretty neat. In other bird species like shrikes and hawks, it has been shown to be used in hunting. And then in blue tits, it's been used in foraging for like berries, and then it's also been seen as a way to attract a mate, and then also like possibly as a way to help them see their offspring. So the current data on color preference, although there isn't much data on chickadees specifically, there is quite a lot of data on blue tits and gray tits. And really good study on blue tits and gray tits showed that they liked silver feeders in the winter and blue feeders in the spring and summer. And it's not that the reason why they like the silver feeders so much in the winter is because, one, silver is apparently a really common feeder color. And also, silver is quite reflective, so it's easy to spot. They can see that. And then blue feeders in the spring and summer is just blue is a very popular color. Everybody likes the color blue, so there's a lot of blue feeders, especially in the spring and summer. There just is. In another study where they tested between dyed, unnaturally colored seeds like blue, pink, and red versus undyed natural seeds, all of the birds showed a preference for the undyed natural seeds, which makes sense because how many times are they going to encounter a bright blue sunflower seed in the wild? Not often. And it should be noted that a lot of the data on color preference is on hummingbirds, which makes sense because hummingbirds like bright colors and just a lot of people like hummingbirds. They're cute. Methods and materials. So I used 8 metal tube feeders and 8 72-inch tall shepherd's hooks, and they were 2 feet apart. I then used Rust-Oleum Painter's Touch, 2 times cover spray paint, and a satin finish to paint these. I used the colors moss green, apple red, summer squash, wildflower blue, blossom white, canyon black, French lilac, and metallic aluminum. And you can see my mouse still. That's the yellow one. You can see colors. Okay. I don't think I need to. Okay. So I painted 2 centimeters below the top. So there's a little mark in here where it's been not painted. And then I also painted 2 centimeters below the feeding port. So you can kind of see one here. This is a feeder port, I think. This one is. Who knows? So I painted 2 centimeters above the feeder ports because I wanted to be sure I didn't get spray paint on the feeder ports. And it worked pretty well. I did get a little paint on the yellow one and the white one, but the yellow and the white paint was really difficult to deal with. And then for food, I just used Royal Wing Black Oil Sunflower Seeds because in a previous study that I did, I found out that they really prefer black oil sunflower seeds, especially in the winter, because they have a high fat content. Data collection methods. So observations will occur within hour increments, and I'm assuming it's going to be between an hour and 2 hours. I'm not going to watch them for more than that. A visit, if they land at the feeder and they take food at a feeding port, and I had this set because in a previous study that I've done, a lot of times some of the smaller birds would just like hang out and chill out at the feeder. Sometimes they would even, I don't know if they were really falling asleep, but they like looked like they were sleeping. They would just hang out, and it's like, they're not making a choice, they're just being lazy. So they have to land, eat, leave. That's a visit. And the birds will be identified and counted on the spot until the amount of birds that come are too high to be accurately counted. Then I will use either a camera or a GoPro or some sort of recording device, and then I will just record them, and then later I will identify them and count them with the recorded video. So here's my feeder orders. So number 1 is yellow, silver, green, blue, purple, white, red, and then black. Then for feeder order number 2, it's purple, black, yellow, silver, red, blue, white, green. And then for feeder order number 3, we've got green, purple, black, yellow, silver, red, white, and blue. And for order number 3, I'm going to switch white and yellow. I haven't used order number 3 in an observation yet, but I also haven't seen any birds yet, so it doesn't matter. Because I realized once I got home and I was uploading my photos that the yellow is always on the left side and it never goes to the right side. So I'm just going to switch them because it's the same with the white. It's never on the left side. So, guys got any questions?
Thank you very much, Emily, for the presentation.
And thank you, Dr. Baldassarre.
And the next person is Luke.
Hi, I'm Luke. I'm going to introduce myself in the presentation that's going to begin, but I would like to say thank you to the Rice Creek Association and all of those who were involved.
Hello, welcome, and thank you all for being here. My name is Luke, and today I will be speaking to you about the determination of the rate of infection of Ophryocystis elektroscirrha upon Danaus plexippus in Oswego County. And now again, without the Latin, we'll be talking about monarch butterflies and their relationship with a parasite. A couple of quick notes before we get started. From here on out, I will be referring to Ophryocystis elektroscirrha as OE because that's a mouthful, and I'm sure me repeat that over and over. Secondly, I want to give a fair warning. I intend on showing you a lot of images of bugs, so I apologize in advance for anyone who's not a fan of bugs. So, what is OE? Like I said, it's a parasite. It's a small protozoan we can see only through a microscope, which you can see here as the little brown spots all over. This parasite uses monarch butterflies as their host and uses them for the entirety of their life cycle. To start, OE is an internal parasite which lives within the monarch caterpillar. They can be transmitted from the parent to the offspring or by ingestion while the caterpillar is feeding. Monarchs use various species of milkweed to deposit eggs and to feed upon, so while visiting the milkweed they can leave behind OE spores which can then be ingested by caterpillars as they feed on the milkweed. During the caterpillar phase, like I said, OE is internal, but upon reaching adulthood and becoming a butterfly, OE becomes visible as external spores, and this is where we can sample and observe the infection rates, which we'll talk about in a little bit. So how harmful is it? That depends on the level of infection. A few spores won't pose much of a threat, but an excessive amount could be life-threatening. A heavily infected butterfly might emerge from chrysalis with damaged wings, a small abdomen, which would indicate poor fat and energy stores, which are crucial to a migrant species, or they may not emerge at all. Now let's talk about monarch butterflies. Beginning from the egg, a monarch butterfly typically takes about 30 days to develop into adulthood. They spend 3 to 4 of those days in the egg before hatching, followed by 10 to 14 in the caterpillar stage, and then another 10 to 14 in pupation. During the caterpillar stage is when they're most vulnerable to predators and when they become infected with OE. And just a little fun thing about monarchs is they feed on milkweed, which has toxic defense chemicals, which they ingest. It doesn't harm them, but what they do is they store them and they reuse them later as their own defenses. So a lot of birds can recognize the monarchs and they don't feed on them because they know they're distasteful and they don't make for a good meal. But anyway, when the caterpillar does become an adult butterfly, it stops feeding on the milkweed and instead starts feeding on nectar from flowers, which is going to be an important part of what I'd like to talk about next, and that's the conservation status and importance of this species. So currently, monarchs have been under observation for listing as an endangered species for a few years now. There is actually going to be a hearing about this and hopefully a decision in December of this year. And so why this species is important is because of their feeding and migratory behavior. Monarchs travel from overwintering sites in Mexico and some southern states all the way up into Canada, and along the way they feed on nectar from a wide range of plants, which helps in plant reproduction. So somewhere along the great evolutionary path, plants began using insects and other animals to assist them in reproduction. And as the plants provided the monarchs with food, the monarchs in turn transport pollen from plant to plant, and the pollen is a reproductive part of the plant. So with such a big migration range, you can only imagine how much assistance they provide, and this makes them a key pollinator, and unfortunately they are not doing well. So this is why we looked into this project and why we did this, because we want to try to figure out what are all the causes of their decline and how much do those factors weigh in. So what we did was we reared caterpillars and we sampled migrant butterflies. So to start, let's talk about the rearing process. Well, we need caterpillars, and that's what we did. We went out and we found them. As caterpillars grow, they go through 5 different levels of growth called instars. We collected them only from a late second instar phase and on, and And this is because we want to get an idea of the infection rate as it happens in nature. If we collect them too young or still in the egg, we might not see an infection because we're rearing them in a controlled environment. By that late second instar, the caterpillar has pretty much gathered as much of an infection as they are likely going to. So after collecting them, we kept them in controlled environments with fresh milkweed, some separation to keep from cross-contaminating, because it's not until the adult stage that the sampling process could begin. Unfortunately, rearing does not always go as planned. Sometimes a caterpillar fails to emerge or emerges with damaged wings, and this might not be because of an illness or OE, it's just a hazard of the life cycle. They need to push themselves out of the pupa, and that level of difficulty changes from individual to individual. There is also a risk of disease. Aside from OE, monarchs are susceptible to different bacteria that can prove fatal. Good news though, we ran into very few problems and most of our reared caterpillars were able to fly free. So, once it did emerge from pupation, this is where we could start sampling. We would record things like wing length, or is it male, female, what's the body condition, and this is also where we took a scale sample to see if OE was present. To do this, we gently applied clear tape across the abdomen, and don't worry, this doesn't hurt them, the tape doesn't actually get stuck, it just removes some scales which won't hinder the butterfly in its day-to-day. We then put the tape onto a microscope slide and we can now look at it and count the spores. So we did find OE spores on some of the raised caterpillars, and this could indicate transmission is occurring from the migrant population. And we also found spores on migrants, which reinforces the idea that the infection is occurring and being transmitted across the whole northern trail as well as the southern trail, which in the southern trail it's expected to be prevalent because this is where the overwintering occurs. So it's interesting to find evidence of infection here because a heavily infected monarch is unlikely to survive or reproduce. So it is worth noting that there is some rate of infection farther along the migration path, even if it is small. We also had some other interesting finds along the way. Typically, as we move into the fall season, the monarchs begin migrating south to their overwintering sites. What was unusual was the reproduction along the way. We collected a few caterpillars late September and early October. This was a late brood and an interesting occurrence to observe. While we're unsure what brought this on, we have started to formulate ideas, one of which could be the environment. As our climate and environment changes, species need to adapt and change as well, so perhaps this is a response to the changing world, and the ever-in-motion wheels of evolution are hard at work. However, it is too soon to make any conclusions. So with this late brood, we also saw an extended pupation. As I said earlier, typical pupation is 10 to 14 days. One of our caterpillars pupated for 23 days, and another we caught in early October finally emerged just the other day after 34 days, which is longer than the typical cycle from egg to adult. So we're wondering now, is this a characteristic of the late brood, or did something else like temperature decline have something to do with it? So some of the problems we encountered along the way. A big one was competition. Monarchs are not the only insects that feed on milkweed. We observed large colonies of milkweed bugs, tussock moths, and some Japanese beetles, which in recent years have been discovered to be milkweed feeders. There was also a high risk of predation, as we found numerous species of spiders inhabiting milkweed, many of whom prey on young monarch caterpillars, and we also found a few assassin bugs, which are often hard to spot because they exhibit mimicry relationship with milkweed bugs. In addition, we saw very little rainfall this summer in Oswego, and with such a dry season, much of the plant life suffered. Additionally, butterflies don't always obtain all the nutrients and minerals they need from plants. Sometimes they visit puddles to kind of splash around and dip their feet to gather certain things, and this is called puddling. So the lack of rain could have an impact on the population, as well as our changing climate. So these are just some images of competitors and predators that I just mentioned. You can see on the top left, we have a cluster of milkweed bugs. We came across countless colonies in our search for monarchs. And to the right and underneath that are photos of tussock moths. which again, we were finding colonies inhabiting the milkweed, and these colonies could be pushing the monarchs out by directly competing with them. The little black ball you see in the middle there is a tachnid fly larvae. And tachnid flies are parasitoids, which means they sort of act as parasites, except for one major difference. Parasites don't intend to kill their host as they need them to survive. Parasitoids don't share that same sentiment. They use their host to reach adulthood and typically don't leave that host in a survivable state. To the right of that, you'll see spiders. There's actually two there. It's hard to see, but if you start counting legs, maybe you can see them both. So I think I caught them mating. Yes, it was awkward, and that's why I thought I'd share it with you. You're welcome. To the right of that, it's a picture of a snail. There were so many snails, more than I've ever seen, and they were just taking over the milkweed, more so than the milkweed bugs. So let's get back to the monarchs. Let's talk about population trends over time. You can see from the data here that the population has been in flux for about 25 years. Now these numbers are gathered from counting clusters at overwintering sites in Mexico. Researchers go out and determine how much area by acre that the monarchs are inhabiting, and in the last overwintering period of 2019 to 2020, they saw a 53% decrease in population, which is a tremendous amount of butterflies and extremely worrying. So what could be contributing to this decline? Well, herbicides and pesticides have played a role in declining numbers over the years. Pesticides specifically target insects, and herbicides fight weeds, which could target milkweed. Both of these things have been used heavily over the past few decades. One of the biggest contributors is habitat loss, specifically in Mexico and the US. So as their habitat has shrunken, so has their population. And you can see here in this photo that this is what a typical cluster looks like at an overwintering site. And finally, there has been some work in habitat reconstruction, but unfortunately, paper wasps have taken advantage of that. In urban settings where people are planting milkweed gardens, paper wasps have basically turned them into traps and are using them as hunting grounds to hunt young monarchs. So what comes next for this project? Well, we'd like to have more data and information to better understand the role that OE is playing in the decline. And while this year's migration has passed us by, we're looking forward to continuing our efforts in the spring and summer of next year. Hopefully, we can rear more caterpillars and catch more migrants to build a larger database. I assure you our work is far from done, and we will continue. Finally, I'd like to give a special thanks to those who have helped in this project. Firstly, Dr. Karen Syme, whose instruction and knowledge proved invaluable. Thank you, Dr. Syme, for all of your help. Secondly, I'd like to thank Gabrielle Waters, who spent a lot of time scouring fields with me. She was instrumental in helping me find caterpillars, and I don't know how to express my appreciativeness enough. Finally, thank you to the Rice Creek Association for funding this project. I'm grateful you chose to provide funding, and I hope I've lived up to your expectations. I thank you very much, and also to all of those at Rice Creek who donated their time to assist with this project. Everyone involved is much appreciated, and I thank you all. So with
All right, thank you. Thank you very much, Luke. The next presenter is Ola.
All right, hello everybody. I also introduced myself at the beginning of the presentation. So again, just like everybody else, I'll just extend a thank you to the Rice Creek Association and we can get started. Thank you. Hello, everybody. My name is Ola Kreszpulska, and I'm an associate professor in the theater department at SUNY Oswego. And today I will be talking to you about my project, which is called The Flora and Fauna of Rice Creek. And the project itself is a coloring book inspired by those two things. And I put a little photo of myself there since I'm just narrating, so you don't get to see me. And you can actually see me working on one of the drawings. Before I jump into the project, I just do find Rice Creek a very beautiful spot. As you can see here, hiking there a lot. Featured is my dog Gumbo. We go to Rice Creek, Fallbrook, and that area, so I was inspired by all the beautiful nature around us. So the project itself, what I wanted to do is, prior to the development of photography, naturalist illustrations were the best means of getting to know the natural world by scientists. In the spirits of these works, I wanted to create a series of illustrations focused on the plants and animals of Rice Creek, along with a corresponding coloring book. Engaging with these illustrations will promote a mindful educational experience rooted in exploring nature. And to capture a subject when you're drawing something, you have to mindfully engage with it. Even if you think you know something and you know what it looks like, to draw it accurately, you have to study it from every angle and take in all of the details. However, a lot of people are intimidated by the idea of drawing and feel like they lack the necessary set. This is where the idea for the coloring book came in. While many people, like I said, are afraid or intimidated by the idea of drawing, they are not afraid to color, which is why we see this boom in coloring books for all ages, including adults. With coloring, there is no pressure to capture the subject in a drawing, but the time spent coloring the subject is still stimulating for engagement, which itself leads to learning. Additionally, just like drawing, coloring is very mindful and can lead to reduce stress. As we all know, outings into nature are great for the body and soul, and bringing a form of them back in a book can continue that feeling, much like a souvenir does from a vacation. By the same thought, recognizing plant and animal species can be a very exciting thing that makes us more conscious of engaging with them in nature. When we thoughtfully study a subject while coloring, it would increase additional outings for further study. So in this project, I started by taking some photographs, and I was photographing both animal species and plant species. Of course, I am an artist, I can draw, but I am not a nature photographer. So I also utilized some existing photographs. So here are some more examples, and these are examples of some plant life. So when it came to the drawings itself, I started by working with various pencils on paper. And once I got through my drawings and got those created, I realized that the pencils, while they looked kind of great in person, were not really scanning into a digital format very well. Again, depending on the thickness of the pencil I used, whether it was something thicker or thinner, they either came out somewhat okay, like the frog, or very, very pale, like the snake. While these can be manipulated a little bit after scanning, I wanted the initial product prior to the scan to look good as well. So I decided to go over those in ink. So after the drawings were created in pencil, I went over each of them with archival ink, either at 0.5 millimeter or 0.3 millimeter, to give me a little bit of line thickness and differentiation. Once the drawings were completed, I then incorporated them into a frame by scanning them. And here's an example of this frog. As you can see, This is now a much increased quality. And again, after scanning it, I am able to manipulate it a little bit in Photoshop as well to bump the contrast and make the lines more pronounced. Again, part of the intention here is so that people can go in and color these. So the lines need to be very visible so that they can color within the line. A couple of more examples here. Here on the left, we have pictured some black-eyed Susans, and on the right, a praying mantis. And again, and this is the example of the snake I showed earlier. So this is what that same rather pale drawing that we couldn't really see very well looks like after it's been enhanced in ink and then scanned again and manipulated in terms of brightness and contrast. I created a time-lapse video that illustrates what the coloring process might be like for one of these pages. So this will take us through coloring the common garter snake. So let me set it up and enjoy. So here you see the finished product and what that will look like after the page is colored in. And the idea is that these will be printed and handed out to visitors at Rice Creek once things like giveaways are safe again. So thank you for your time, and I hope you enjoyed this presentation.
Thank you, Ola. That was a great job. The next presentation that we have is Miryam.
Hi, I also introduced myself in my presentation, but I'd like to thank Rice Creek and Dr. Coon.
Hello, my name is Miryam, and this presentation is about our research on the effects of atrazine on the eastern elliptiocomplinate, more commonly known as the freshwater mussel. To start off with, what is atrazine? Atrazine is a chemical compound belonging to the triazine group. It is a commonly used herbicide throughout the United States and the world. It works by inhibiting the photosynthetic activity inside the chloroplast membrane. In the United States alone, around 76.4 million pounds are used annually. Because of this, it's been detected in the ground as well in both surface and drinking water. Atrazine is moderately hydrophilic and has a half-life ranging up to a year. This means it's able to dissolve in water and it stays there for a very long time. This also means that the organisms that live in the body of water are constantly exposed to atrazine and for long periods of time.
So, taking a look at previous studies on atrazine, there have been several studies on its effects on animals. Some common findings were that atrazine affects the endocrine system. It has been shown to reduce production of GH and affects the normal function of the reproductive system. It's also shown to contribute to development, causing birth defects. Humans have been shown to have a higher susceptibility to birth defects, and we have an increased risk of breast and prostate cancer when exposed to atrazine. So, what do we hope to find out with this study? Our goal is to find the effects of the chemical atrazine on the metabolism of different tissues, and we do this through looking at mussels. Mussels are aquatic animals and are constantly exposed to atrazine since the common water pollutant. We are doing this through histology, so sectioning and looking at the mussel tissue as well as the cells closely under a microscope. The second part of our research is measuring the bioaccumulation, or the amount of atrazine that accumulates in the cells of different mussel tissue. Ultimately, we want to see the difference between untreated mussels and those that have been exposed to atrazine, and then measure how much atrazine in the tissue led to this. So why do we choose mussels? We chose freshwater mussels as our model since they're commonly used as biomarkers for toxicology or teratogenicity testing. This because mussels are filter feeders, and as a result, anything they're exposed to is absorbed directly into their tissue and ends up in their cells. They are constantly exposed to atrazine in their natural environment since they are aquatic animals and atrazine is a common pollutant. Even though there are some studies regarding the effects of atrazine on behavior of freshwater mussels, there's yet anything to be reported regarding the effects of bioaccumulation of atrazine and cell metabolism. Since there's already studies focusing on vertebrates, results from invertebrates such as a mussel would be significant. So how and where did we get the data? It all started at Rice Creek Field Station, from where we collected the mussels. These mussels were brought to Shinneman, where, as seen in the topmost image, 2 tanks were set up, one as the control and one as the atrazine treatment tank. Other than one tank receiving atrazine treatment once a week, all other conditions remained constant. The temperature was held at 15 degrees Celsius, the lights were on a 12-hour cycle, and the mussels were fed every other day. The culture period was 4 weeks, and during this time, the tank was cleaned once a week, and the water chemistry of pH, ammonia, nitrate, and nitrite concentrations and water hardness were measured. Following the 4 weeks, the mussels were dissected into the gills, the viscera, and the mantle. The dissection process is shown in image B, and the different parts are shown in C. Half of these organs were used for bioaccumulation studies, while the other half was used for histology. At this point, we had the mussels broken up into parts, so what next? We placed the tissue in wax and cut it very thinly using a microtome. We then stained it using HE and in some cases tunnel. The histological analysis was completed by a previous researcher, and it was found that there were histopathological events on the gills and the gonadal tissue. We will actually be looking at those slides later in the presentation. We then wanted to confirm the results, so we wanted to see if it was reproducible and to conclude on what was actually observed. In order to do this, we continued the histological analysis on all the replicates of the control and the treated samples. There were 4 control mussels and 8 treated ones. Through this, we have proven that the teratogenic effects seen in the gills and the gonad tissues were significant in the treated mussels compared to the control. However, interestingly enough, there were no histopathological effects seen in the mantle tissue, only the gills and viscera. So what were we able to find out about the tissue from looking at it through a microscope? One thing we found was that the ciliary epithelium of the gills was affected by the atrazine. So looking at the slides more closely, the control is on the left and it's A to A double prime, and the atrazine treated samples are on the right with B to B double prime. All of these slides are biological replicates, so they're all images of different mussels. First, let's look at the long squiggly projections that you can see in the control. Notice how they are actually pretty thick. That's the epithelial layer. Now at the atrazine treated, notice how it's very thin and the epithelial layer has been disintegrated. Next, look at the little hair-like projections coming off in the control. These are all either fused or missing in the atrazine treated, and this is because the epithelial layer has been disintegrated, so they have nothing to attach to. Next, look at the skeletal rod. So in A prime, it's denoted as SKR. And notice how there's a nice rod there. In the atrazine treated samples, that is missing, and it's very important for the structure of the gills. It's kind of like the bones in the human body. Lastly, the ciliary epithelium, so the long squigglies we looked at earlier, are arranged in a row, and they're all uniformly spaced, and the size of the cells are all consistent across the tissue, and this isn't the case with the atrazine treated. These effects that were observed with the initial histological analysis were reproducible, and this concludes the teratogenetic effects of atrazine on the gills. Now, looking at the gonads, the reproductive system, the atrazine was able to introduce structural abnormalities in the ovarian tissue. The control panel is the topmost one from A to A double prime, and then atrazine treated B to B double prime and C to C double prime are seen below it. These are all images of the same muscle section, they're just more zoomed in each slide. So first, let's take a look at the ovarian acini, so at slide A prime, and that loop, it's very thick, it's very healthy looking, it has a lot of particles in it, and that is missing in the atrazine treated samples of B and C. Also, looking at A prime, you can see the stalk, it's noted by the S, it's found on the middle left of the slide, and notice how thick it is. And another slide was found over on B double prime, and notice how thin it is. It's not really able to connect to the acinifollicular wall, which has already also been damaged and disintegrated. So overall here, we can see the disintegration of the ovarian acini, the oocyte, the nucleus, the ovarian follicles, and the epithelium of the genital ducts. We can also see cytolysis inside the oocytes due to damage in the yolk and that stalk connected to the acinifollicular wall. So overall, this ovarian tissue has been highly damaged. Next, let's look at the male reproductive organ. So again, on the left are controls from A to A double prime, and on the right are the atrazine treated samples of B to B double prime. All of these slides are the same slide, just more zoomed in and in different spots. So looking at it, you can see that there's a decreased number and condensation of spermogenetic cells. So looking at A prime, you can see lots of different purple dots all throughout it, and they're very well defined. And looking at the atrazine treated B prime right next to it, if you zoom in, they're all condensation, they're not very clearly defined, and there's not as many of them. We also see the appearance of intratubular vacuoles and necrosis of the gonadal ducts and spermocyte. So just like the females, the male reproductive system has also been inhibited. We wanted to see what mechanisms were behind the differences seen in the gills. So we used a parallel section of the same specimen for an apoptotic assay, and using a double-stained immunohistological method. So for this, we used two antibodies, DAPI and tunnel. DAPI is seen in all of the blue, and it is able to show all the nucleus. So every living cell is denoted in blue. And we also use the antibody tunnel, which shows all of the dead cells, so anyone who went under apoptosis. Now in the control, which is on the far left, you can see that there's very little green, so very little dead cells, and they're mostly limited to the ends. And this is completely normal to have some apoptotic cells for the normal function of the cell. In the middle and far right, we have B and B prime, so these are the atrazine treated. And you can see how there's a lot more green, so there's a lot more cells dying. And you can see that it extends all the way down the rod, so structural aspects are lost. Lastly, you can see apoptosis in the ciliary epithelium as well as the ciliary cells. So this shows the fusion of the ciliary cells, especially in the frontal cilia, so FC. It's on the far right of the panels B and B prime. So ultimately, what this image shows us is that atrazine induced apoptosis in the gill slits. This image shows atrazine-induced apoptosis in the inner gill lamellae. So all of these show the same region, just under different wavelengths. So in A, you can see all the apoptosis happening. A prime shows all the living cells. A double prime shows any mistakes, so any red blood cells, dust particles, things like that, that would show up as apoptosis but actually aren't. And as you can see, there's only 4 spots in the right part of the A double prime slide that show this. And so when counting the apoptosis, we can just negate those. A triple prime shows all 3 of these, images put together. So what we can understand from all this is that atrazine is inducing apoptosis in both the inner gill lamellae as well as the gill filaments. As mentioned earlier, there is a second part to the project, and that is bioaccumulation. This has been done through ELISA testing and the LCMS instrument. We do have a standardized protocol for bioaccumulation with the LCMS. In order to identify if any atrazine contamination was at Rice Creek, which is where we got the mussels, we used an ELISA test, otherwise known as an enzyme-linked immunoabsorbent assay. So the way an ELISA test works is there's an antibody immobilized on the plate. After that, a standard is added, in our case atrazine, and after all the extra standard is washed off, a substrate solution is added to give it a color signal. The intensity of the color is inversely proportional to the concentration of atrazine present. Water samples were collected from 4 different sites at Rice Creek Field Station, and you can see where that was with a little pinpoint in the top left image. Using an ELISA test, the concentration was found to be less than 1 nanogram per milliliter. So at such a low concentration, atrazine is not reported to have any effects on aquatic or other organisms. So, ongoing studies. The LCMS protocol has been standardized and it is listed below, and we're currently working on the analysis. So, in conclusion, atrazine affected lots of different aspects of the mussel. We looked at the gill phenotypes and we noticed that the size as well as the arrangement of the ciliary epithelium was affected. As a result of this, the cells attached to the ciliary epithelium were also getting affected, and so they were either missing or getting fused together, and this means that they're not able to function properly. In addition, the skeletal rods, as well as the tissue cells around it, were reduced in both number and size. And the reason behind this have been identified as induced cell death. So all cells that were affected by atrazine were found to be undergoing apoptosis. Inside of the gonads, the reproductive system, we did see a reduced number and condensation of spermogenetic cells, and we saw necrosis of the gonadal duct system. There was also disintegration inside the ovarian acini and the cytolysis inside oocyte observed. So both the male and the female reproductive systems were affected. Taking all of these different things into account, normal metabolism would be affected, thereby preventing normal function of these mussels. We would like to thank Dr. McKenzie for helping us with collection and maintenance of the mussels in lab conditions. We'd also like to thank Dr. McElwain for providing valuable comments on data analysis, Dr. Nimagata for helping with histological and ELISA analysis, and Dr. Geary for helping with LCMS instrument and bioaccumulation. Finally, we'd like to thank Rice Creek Associate Grant, Challenge Grant, and the NSF Small Grant for funding the project. Thank you.
Thank you, Miryam. That was a great presentation. Now, the next presenter is Shawn.
Hi, everyone. I'm Shawn Geary. I'm a senior biology major, and I'd like to thank Rice Creek, Dr. O'Lory, Dr. Hammerly, and Dr. Sard. So I hope you all enjoy. Hi, everyone. I'm Shawn, and I'm going to be talking about a continuation of a long-term survey of Rice Creek amphibians for the prevalence of 2 global pathogens. So I'm going to start off talking about declining amphibian populations. Since around the 1980s, amphibian populations have been steadily declining, substantially actually. Amphibians are thought to be one of the most at-risk vertebrate groups to population decline, with around 70% of amphibian species thought to be susceptible to declines happening to their populations. There's really multiple factors that interact to contribute to declines, with the major ones including climate change, habitat loss, pollution, and what I'm going to be talking about today, which is enigmatic declines. An enigmatic decline is a mass die-off event in a population for which the cause was originally unknown because they were happening in areas where amphibian habitats were really untouched and there wasn't really a heavy human presence in that area. But as scientists really looked into it further, they found that the individuals in populations that were declining were infected with either one of two diseases, or one of two major diseases. The first being chytridiomycosis, and the second major disease affecting amphibians is ranavirosis. Ranavirosis is a disease caused by multiple different strains of viruses from the genus Ranavirus, RV for short. It's a double-stranded DNA virus that is spread through direct contact of an infected individual or through soil or water if the virus is released. It can be detected in reptile, fish, or amphibian groups. Ranavirus can target many different organs within amphibians, and it really depends on the strain which one it focuses on or it infects in the amphibian. But the liver is the most common target for infection, with the spleen and kidney being other common targets. Skin ulceration and lesions are really noticeable in individuals as the infection progresses, and in dead individuals from it, it's a giveaway that ranavirus probably caused its death. If you look in the figure in the lower right-hand corner, part A shows an adult frog with red right on its legs, and that is skin lesions and internal bleeding from the virus. And the picture B shows a tadpole which had ranavirus, and you can see it just has lesions and bleeding all over it. Relating to that is that amphibian larvae are the most susceptible to ranavirus because ranavirus can cause death in an amphibian larvae in under a week, which is really shocking. Ranavirus is known to infect at least 72 amphibian species, though some species have shown resistance to infection. If you look in the top right-hand corner, you'll see all the different countries around the globe that have reported ranavirus infections. And this figure shows just the prevalence of ranavirus at Rice Creek. The y-axis shows infection prevalence of ranavirus, and the x-axis shows the year. It's really fluctuated throughout the years with a low infection rate or low infection prevalence occurring in 2016 with about 10% infection. Chytridiomycosis is caused by the fungal pathogen Batrachochytrium dendrobatitis, or BD for short. But it was also found recently, in around 2015, that there is actually another strain, Batrachochytrium salamandrivorans, or B-sal for short. BD is the strain that is found in the Oswego area. It has two main life stages, a mobile zoospore, which can survive in the environment for up to 3 months before it infects an individual. And then once it does so, it matures into a sporangium, which grows new zoospores inside it until they are eventually released. And then they can infect either the same individuals, but different skin cells on that individual, or go on and infect new individuals. It's really of note that BD infects the skin because of how important skin is for amphibians. Amphibians use it for gas exchange and for the exchange of water. So if the infection spreads far enough throughout their skin, it really impedes this and stops them from being able to do these vital functions, and eventually it results in their death. Chytridiomycosis is associated with the declines of over 200 species of amphibians. But similar to ranavirus, there are also some species that are resistant to it. And if you look at the map to the right that shows the Americas, the area in red is where the environment is suitable for BD to spread. And all the red bullet points are areas where BD has been recorded in amphibian populations. And this figure shows BD prevalence at Rice Creek, again with the y-axis showing infection prevalence of BD and the x-axis showing the year. Similar to ranavirus, it fluctuates throughout the years, but it fluctuates at different years than ranavirus did. The lowest infection prevalence year was 2013, where it only infected around 3% of the population, or 3% of the amphibians that were sampled. So how do we sample these amphibians for ranavirus and BD? So this has been a little difficult because of COVID-19, obviously, because of social distancing and the need for masks, but we have thankfully been able to still get sampling done. This year we've been able to sample around 96 specimens, although we still do run into a little trouble with it. So part of our sampling requires us to go out and so we look through the environment for the amphibians. We look in places like vernal pools, Rice Creek Pond, Rice Creek, under logs, under rocks, throughout the woods around Rice Creek. But once we actually have the amphibians, we need a team or a pair of people to take the data and weigh them and identify them and measure them. But with social distancing, that can be a little difficult, obviously. But thankfully, 2 members of our group that sample live together, so we have been able to have them do assistant stuff without worrying about social distancing. Other things we record are water quality, so like pH, and we take a toe or tail clip from an amphibian or a frog, and that allows us to test for ranavirus, and we take a skin swab, and that allows us to test for BD. So the molecular side has been a little more troublesome than the actual sampling side. COVID-19 really prevented us from getting into the lab to extract samples last semester. And another issue with that is that the professor who was once in charge of doing the molecular aspect of things moved on to another college, or another job. So we've been trying to really get it up and running again on the molecular side, but because these samples are so small, it's been giving us a bit of trouble. If you look at the bottom right-hand corner, that is actually the average size of a toe or tail clip, and there is a toothpick for scale there. So that tiny little black dot is the sample size with the toothpick next to it. So it's very small, but it's definitely still enough for us to extract DNA from it. So right now we're really just trying to refine that technique and get it so we can consistently get enough DNA from it. And then after that, we can then move on to the next part of detection, which is PCR, where we copy the DNA, and gel electrophoresis, which allows us to really diagnose if BD or ranavirus is present in the specimen. So for possible future research, I'm really interested in starting to do environmental DNA or eDNA research with BD and ranavirus in our area. It has been found in a couple papers that eDNA can be used to predict infection prevalence. So Hall et al. 2015 was able to predict ranavirus infections, and Kamaroff and Goldberg 2017 was able to correctly actually predict a die-off event due to increasing eDNA concentrations of BD in an area. So for Rice Creek, the first thing we would do is see how sensitive an eDNA assay for BD or ranavirus would be. And then once we establish the sensitivity, we could then start comparing the physical sampling results to the eDNA results to sort of see if they correspond in any way. And here is my literature cited. Thank you for listening to my presentation.
Thank you very much. And last but not least, we have Sydney.
Hi, my name is Sydney. I'm actually a recent graduate. I just graduated from SUNY Oswego this past May with a biology degree. I've been doing research with Dr. Sard throughout my spring semester until, unfortunately, COVID hit. So, but thankfully to Rice Creek, we were able to pick the project back up throughout the summer, and this is what I've been working on for the past 4 months.
Hi everyone, thank you for coming to my presentation. My name is Sydney, and today I'm going to be talking about environmental DNA and the relative contributions of intracellular and extracellular DNA and how that applies to the North American river otter and its detection rates. So what is environmental DNA? Environmental DNA, or eDNA, is any DNA that can be sampled from the environment, typically sampled from water, sediment, snow, or soil. For my project, we sampled water samples. These don't require the target organism. Sources of common eDNA include urine, feces, saliva, or skin cells, all left behind by the organisms. And we can use eDNA to infer a species presence in the recent past or the general area. So some background on my target species, the North American river otter, or scientifically known as Lontra canadensis. They are semi-aquatic animals. They like to live in isolated areas away from high human activity. They're similar to beavers. There have been studies done that show a correlation between the number of beaver flowages and also the use of watersheds by river otters. And they like to spend most of their time in the water or on shorelines, and they can also make burrows and lodges and dens like beavers do. There are also found all throughout the continental US and Canada. So a little bit about their anatomy. They have a very valuable fur pelt. They don't have an insulating fat layer, so their fur pelt is what keeps them warm because they spend a lot of their time in cold water. They are great swimmers, and their large lung capacity and webbed hind feet help with this. Right now, they have stable population numbers across the world, but they do face several threats, and some of those threats include hunting and trapping, logging, water pollution, and habitat destruction due to residential and commercial development. They currently are protected under CITES Appendix 2. This regulates international trade involved with the species, and this is essential to maintaining stable population numbers. So understanding river otter distributions can help improve restoration and conservation purposes in response to these threats and how they change over time as a result of climate change, habitat destruction, urbanization, and coastal development. And by understanding the ecology of eDNA a little bit better, hopefully we can use the information and knowledge that we've gained from this project and use the river otter as a model to understanding the distributions of their endangered counterparts like the southern river otter, hairy-nosed otter, giant otter, and India smooth-coated otter. So our goal was to extend the field's knowledge associated with the ecology of eDNA. That involves things like where it originally came from, how far it was transported, or how long it's lasted in the environment. And we were going to apply that to our project by determining if the detection rate of a target organism, which is the river otter, differed based on the state at which eDNA was collected. So that would be whether it was intracellular versus extracellular and whether that was affected. And we were going to compare that between 3 different locations at Rice Creek. So these are just the 3 locations that we sampled from. We sampled from above Rice Creek Pond and downstream of Rice Creek Pond, and also from right in the pond itself. So as a brief overview for our protocol that we came up with for this project, we took our 15 milliliter water samples from the field, we brought them back to the lab, we popped them in the centrifuge real quick for 10 minutes, and this enabled the separation of intracellular and extracellular DNA. So this is kind of a step-by-step of the protocol that we came up with using a CTAB DNA extraction. Overall, we made this protocol to be a cheaper alternative compared to the usual QIAGEN extraction kits. From this experiment, we determined that it is efficient and effective at extracting DNA, and roughly it costs around $3 to $5 per sample to extract compared to using a QIAGEN kit where it costs upwards of $15 per sample. So for this protocol, we use a precipitation with sodium acetate and ethanol, a digestion with CTAB and proteinase K, and then a extraction with chloroform and isoamyl alcohol. So after successfully extracting our DNAs, we use a nanodrop to measure DNA concentration. So on the y-axis, we have our eDNA concentration, and the x-axis are the 3 locations that we sampled from. So overall, the median intracellular DNA concentration was significantly higher than extracted DNA concentration from extracellular samples, and the highest for both intra and extracellular concentrations were found in the pond samples. So for our in silico analysis, we used a published qPCR assay by Paget Stewart. We adapted that to an endpoint PCR, and through using primer blast, we compared that to different related species to the river otter, but there wasn't enough data in the database to sufficiently compare. However, we did get enough information for the marten, and we found that there was 3 forward and 4 reverse mismatches in the target template found for that, but there was no target templates found for the fisher. Our expected base product was 147 base pairs, and then we use this information to move on to our in vitro analysis. So for our in vitro analysis, we had different samples of positive controls for the river otter, marten, and fisher, and since we adapted our assay from a qPCR to an endpoint, we had to ensure that it was species specific. So that means we had to make sure that only would amplify river otter DNA. So when we used that and applied that to our samples, that we knew whether there was a presence or absence of it, and that it wasn't some other species, some other related species like the marten or fisher. To do this, we had to vary the cycling conditions a little bit. We increased the annealing temperature to 66 degrees Celsius, and we also decreased the number of cycles from 44 to 30 cycles. So these are some of the results of our gels from our in vitro analysis. Figure 4 is our initial assay. You can see that there's amplification across all species, including M. pinanti and M. americana, which are the fishers and martens, and we needed to eliminate that. So that's why we modified the annealing temperature. We can still see a little bit of nonspecific amplification in the non-target species. So that's why we decreased the number of cycles, and then we got rid of that nonspecific amplification, and we're only amplified our river otter positive controls, and there was no contamination since our NTC was clean. So before moving forward and applying this assay to our samples that we extracted, we wanted to make sure that inhibition wouldn't be a problem, and we hoped that the CTAB buffer would eliminate potential inhibitors in the PCR. So to make sure of this, I randomly chose 2 intracellular and 2 extracellular samples from each location. I took 4 microliters of eDNA and combined that with 1 microliter of positive control river otter DNA in another tube. I made a 1 to 10 dilution and a 1 to 100 dilution of each sample, and so what we were looking for is if inhibition was a problem, then we would see increasing band intensity as the dilutions got greater, because that means that the pure sample of it was being inhibited, but the inhibitors were not able to act on our more diluted samples, but that is the exact opposite of what we ended up seeing. So this is that gel from the inhibition experiment. As you can see, the band intensity decreases as the dilution gets greater, because there's less of that positive control DNA in the diluted sample. This was consistent throughout all of our samples, and our NTC was clean, so we were able to conclude that inhibition wasn't a problem, and we could move forward with our in vitro analysis. So after applying our PCR assay to our extracellular and intracellular samples, we saw little to no amplification across all samples. So to increase our chances of detecting river otter DNA, we decided to increase the number of annealing cycles to 60, which would double the amount of DNA being amplified. Even though increasing the annealing cycles also led to some nonspecific amplification again, we were able to use the expected base pair at 147 and positive control samples to safely determine that there was a presence of river otter DNA. So this figure demonstrates the proportion of samples that had detections of River Otter DNA, with our extracellular samples on the left and intracellular samples on the right. The darker blue bar is the proportion of samples that had a detection, yes. The upstream samples did not demonstrate any detections for either intracellular or extracellular. For both types of DNA, there was no significant difference between detections of River Otter DNA in samples collected in the pond versus downstream, but there was a significant difference between the detections found in the pond and upstream, since there were no detections in upstream samples. Overall, there wasn't any significant difference in the total number of detections found in either types of DNA when compared to each other. From this, we can conclude that our differential centrifugation protocol through a CTAB DNA extraction was efficient at extracting DNA and more specifically extracting our target organism DNA, the River Otter. So environmental DNA is still a pretty new tool in ecology, and its uses for how it can be applied to conservation are still being explored, but pursuing research in this field can improve our accuracy of detecting rare species and analyzing species populations that are at low density. From here, environmental DNA can further research into new conservation efforts that aim to assess species distributions, especially those of endangered and threatened species, and hopefully this can also help improve the information available on river otters and their DNA and apply that to conservation research and efforts for their endangered relatives. So I wanted to say thank you to Dr. Sard for helping me out with this project, and also another research student, Lily Pavward, that helped me do some of the extractions and sample collection. And also, of course, Rice Creek for funding this project, and thanks for coming to my presentation.
All right, thank you very much, everybody. All the presentations were great and all your job is amazing. Now we're moving to the Q&A session. So I asked the participants that want to ask a question to please raise your hand. If you raise your hand, then we can call you out, and the person that is going to answer the question can answer for you. You can also use the Q&A chat underneath. So if you don't want to talk, you can ask your question there, and the person will be able to answer for you. Any questions for the attendees? We have a question for Elizabeth. Elizabeth, you should be able to unmute yourself and ask the question. Elizabeth, can you unmute yourself?
Okay, maybe you want to move on.
As we figure out somebody else with a question.
Maria, there's a question in the chat.
Yeah, I have the question in the chat.
Okay. Oh, but you can talk now. So you want to ask the question?
Oh yeah, I was asking what happens when you mistakenly add the gel to the control group.
For whom is this question?
Oh, the last person who just presented.
Okay.
I'm sorry, what do you mean mistakenly add a gel to the control group?
Is it like how the control group, I think it's either the dependent or the control group. Instead you add something gel to it, and I was asking that what happens if you mistakenly add the gel surrey? What will happen?
Are you talking about for the inhibition experiment?
Yes.
So for that experiment, we we had to run the experiment a couple times because we were having some problems with our samples amplifying. So we had some problems with our reagents, but once we cleared up which reagents were faulty, that experiment that I showed, the final gel picture, that was our final experiment, and we didn't see inhibition in our samples that we added the positive controls to. So the positive controls are to our known River Otter samples. So you know that that DNA should amplify at that expected base pair. So when you run it through a PCR, as long as you know that your reagents work properly, you should be seeing bands when you look at your gel. And that's kind of what that experiment was showing, is that the samples that had known River Otter DNA in them were the intensity of the band like decreased, and then the NTCs are the control. So those should always be blank. There should never be any amplification in the controls, because if there is, that means the samples got contaminated somehow, and you always want your NTCs to be clean. I'm not sure if that answered your question, but hopefully that helped a little bit about the controls.
Yeah, it does.
We have a question from Kamal.
For Sydney, can you explain the difference between the intra and extra DNA, and how do you detect them in the sample?
So the intracellular is what's contained like in the whole cell itself, and extracellular is kind of just like free floating fragments in the water. So we actually expected to see more intracellular because they're cells itself, like whole cells. We separated them through centrifugation. So when we took the samples back to the lab and we put them in the centrifuge, I think it was for 10 minutes immediately, we poured off the supernatant, which should contain the free floating DNA, into another tube containing the sodium acetate and ethanol to precipitate that DNA. And the pellet, which should be stuck to the outside of the centrifuge tube, should contain the intracellular DNA. And since we were able to see DNA concentrations in both our extracellular and intracellular sample, then we were able to conclude that our protocol was sufficient in separating them through that process.
Thank you.
Any other questions for any of the panelists?
I think Dr. Helquist had a question for me. And according to the EPA, I believe they have 10 parts per billion as their safety mark-off. And so Rice Creek is far below that. We found it at 0.9 parts per billion, around there. So we're good there. And then in the actual experiment for the treatment that we gave to the mussels, I think we did go slightly above the EPA recommendation of 10 parts per billion for their water concentration.
I'm sorry, but I have to ask this. What are the independent and the dependent variable of your experiment?
For me?
Yes, please.
So we compared how atrazine affects mussels. So we had a control sample set up where the freshwater mussels were in water that had a negligible amount of atrazine in it, so very low concentrations. And then we compared that to atrazine treated. So we had slightly above, I think, 10 parts per billion of atrazine in that tank. And then we took the two mussels and we compared the histologicals. So we looked under the slides and looked at differences between the two. And we're also doing currently bioaccumulation to see how much atrazine caused the effects that we're seeing under the microscope.
Okay, thank you.
Yes, I have a question for Ola. So you know when your coloring books are going to be available at Rice Creek?
I have the drawings. I need to send them to Kamal, who is going to help with printing. So I'm not sure of that answer, but I don't know when stuff is safe to give away in terms of COVID. Like, I feel like right now it's probably a bad idea to give things away. I don't know that.
Whenever you're ready, we'll print it.
Okay, thank you.
Yes. And may I make a comment?
Mm-hmm.
I really like your ideas. It's going to help. I mean, it's going to be an asset for us because we have a lot of children who can use those. But also, I want to make a comment. John Weeks, one of the professionals who established the field station, was here last week, and we worked with him in the building, and he talked a lot about his paintings. He has paintings all over the field station. And one of the things he mentioned, he said, whenever he feels stress, he will go and paint. And he said, it's just did him a lot of good. And then all of a sudden he said, I hope it did not take me away from my family because I work a lot on it. So does painting take a lot of your time away from your family?
From my family, no, but it is a very good distressor. I will say that. Like, drawing has been so good for just everything happening right now. And I mean, I think the first group that was talking, and the counseling group, like, nature helps, but then drawing combined with that, it was very therapeutic for me. So I enjoyed it.
And then just one comment then, and then actually I don't have anything else. For Ellen and Cory, I think also that your work is really good and it's gonna bring more students and more people to the field station. I just wanna know actually your opinion on the response, students' respond to your sessions.
All of the students that have come have just really loved it. The ones that have never been to Rice Creek before are just really surprised that they have access to this, that it's part of SUNY Oswego and it's something that they can go to. They're surprised that a shuttle bus even can take them there. We were surprised when I first, posted that for the freshman students. I'm like, well, I'm probably going to get a lot of bio and zone majors who come, which we did, but we also got a lot of other majors, broadcasting, journalism, business majors. It's just, they ran the gamut, and the response has been really, really good. Ellen or Kyle, do you have anything?
Yeah, I absolutely agree with what Cory says. I think when the students come out there, some of them don't necessarily know what to expect. And after doing some of our guided meditation walks, they just are pretty amazed at what happened in that half an hour that we spent with them. And many of them actually came back for more of them and have said that now they'll continue to utilize Rice Creek.
Thank you.
I think Emily is going to answer a question.
Yeah. So when determining the feeder order, I just actually put the different colors in a random list generator online, because when I tried doing it myself, my orders weren't there wasn't much variation. And the reason why I had the feeder orders in the first place is because if I was to just have the same order every single time, we could see a preference for certain colors, but it would more be a preference for certain feeder locations. So there could be a preference for feeders at the end, because at both ends there were some shrubbery. And because chickadees are small birds, they are a prey species. And like when I was setting my feeders up last Wednesday, I actually did see 2 hawks flying around looking for a meal. So it just prevents our data from showing like they like the feeders based on location rather than color.
I think Kristin wants to answer a question.
Yeah, I had a question for Luke. Luke, you mentioned something about, I don't know if I have the wording right, something about a northern migration route and a southern route or something like this. Could you explain what that is?
Yeah, when I was talking about a southern route and a northern route, they're part of the same route. It's just that the more southern area is the kind of overwintering. In the southern states and down in Mexico is where they overwinter, and they migrate up past us into Canada, and then in the fall they go back south.
So you were saying you were expecting, or maybe you find more of a parasite load in the south because the ones that are heavily parasitized don't make it as far north? Is that right?
Right, because they're overwintering down there, where you're probably going to see more of a population, and heavily infected monarchs, if they can fly at all, they're probably not going to make it very far. So a lower infection would be expected to make it this far if it made it at all.
Laurel,
yeah. My question's also for Luke. Couple things. I have planted a whole lot of milkweed in my backyard for the purposes of helping the butterflies. In fact, I brought some of them over to Rice Creek to put in the container this summer. But what did you mention about, was it the wasps or something are increased because of that?
Paper wasps. It's something I read about recently. In more city kind of areas where people have been planting milkweed gardens, the wasps have been utilizing that as hunting grounds.
Okay, all right, well that makes sense because we have a lot of those as well. And then I was curious to see the other pictures of the other predators because I could definitely say, oh yeah, saw that. So all right, we'll be wiser to watching out this time next year. Thank you.
Yeah, actually, Luke, you mentioned some of the factors which affect the population or the monarch population, and you did not list the OE among those. So how common is that?
Because right now it's really unsure how much of a role they're playing. It's a question of, are they causing population decline, or are we seeing more infection rates because of the population decline? And so they have, rather than the infection being spread across a large population, it's being spread across a small population. So part of it was just trying to figure out how much of an infection rate there is and how much of a role it's playing.
But there is nothing in the literature about its effect?
For our area, I don't know if it's playing much of a role because the infection rate is very low here. And I know in some southern states where they're doing work with this, it's playing much more of a role, and they think it's much more suspect to being a more prominent cause of population decline than it is up north where we are.
Thank you.
Ola has a question.
My question was kind of for the presentation on amphibians, but also for anybody who was collecting and analyzing sample. How is that process different during COVID, where you can't like stand next to a partner or have somebody help you with something? What were some challenges with that?
So for sample collection, it was not quite as difficult as you would think because for the most part, we're kind of spread out just looking around for amphibians. But when we actually had to do measurements, we do like a team that does the measurements. And luckily we did have like a pair of people on our team, like Luke. Thanks, Luke. Who lived together, so there wasn't a concern for social distancing for them. So I'm very thankful that we had them on there. But for running samples in the lab, basically just masks and trying not to be in the lab as much with as many people in there. So.
Kristin?
I had a question for Emily. Emily, you said that, I guess past studies had, conjectured that the feeder preference in some of the related bird species was kind of related to what was the most common bird feeder color that people were putting out. So is your hypothesis related to that for the chickadees? Are you expecting it to be related to something about their vision or something related to how common that color is in people's bird feeders?
Originally I wanted to do something where I would have members of the community just poll what their favorite feeder color was and then compare that to my data. But also I originally was going to analyze the UV components of the paint, but I emailed the company and they never got back to me. So I guess I'm just not gonna do that anymore. But I'm assuming it's going to be the favorite color again, because the study that was on blue tits, they're really closely related to black-capped chickadees. So it's very likely that we're gonna get similar data.
Any more questions for any of the presenters? Hearing none, I guess we'll go to Laurel for the closing remarks.
So I'm Laurel Arts, and I'm the president of the Rice Creek Associates. And I just want to say congratulations to all of our presenters for sharing their important work. And thank you to our audience for attending and showing support for our small grants awardees. Hopefully you learned something new today and have a good idea of how important the research opportunities the field station offers are to the college students, the faculty, and the college community. Also a big thanks to Maria for coordinating the program, and also to Kamal and Kristin and Wendy, who we hope is in attendance, for all the things they do to help us out. And the Small Grants Program is one of those important efforts. Since 1996, RCA has funded over 100 projects totaling $132,000. Due to the financial constraints at this time, throughout the SUNY system, this year we find ourselves in a position of needing to find additional sources of funding to allow continuation of this important research program. You can help by becoming a member of RCA. Our membership fees are extremely reasonable. There is a form available on the RCA link on the Rice Creek page of the oswego.edu website, or I can make it simple. If you just email me at rca.oswego.edu, I can send you the form. There is also a brochure that's available at the station, but we can't get you there right now. But if you email me, I can get that information to you. So you can use that to enroll as a member of RCA. You can also use that same form to make an additional donation if you would care to towards the research that we do for the Small Grants Program. So we hope that you will consider that. And again, thank you so much for showing up today and showing your support for our researchers. We really appreciate it.
Thank you very much, everybody.
Laurel mentioned Wendy. Wendy's home. She's doing all right. She was here for the first two presentations, and she went to rest. So please feel free to give her a call or a text message.
Oh, it says she's back watching.
Oh, okay. She's back watching, all right.
So, yeah. All right.
Thank you very much for coming. Go and enjoy the nice day outside.
All right.